inside your pantry, probably sitting in one of those little plastic bears, is a substance that hospitals are actually starting to use to kill antibiotic-resistant superbugs.
Right, which sounds like science fiction, but it's really happening.
Yeah, it's wild. But here's the catch. For you listening at home, the medicine inside that bear is really only as good as the diet of the insect that, well, threw it up.
Yeah, that is a very visceral way to put it.
I mean, it's true, right?
Oh, it's entirely accurate biologically speaking. I mean, we tend to strictly categorize our world, like food goes in the kitchen pantry and medicine goes in the pharmacy cabinet.
Exactly. We draw a hard line.
Right, but honey, it just completely blurs that boundary. It's this highly engineered bioactive substance.
Which perfectly sets up our mission for this deep dive today. We are looking at a pretty fascinating 2023 research article from the journal Discover Sustainability.
It's a great paper.
Yeah, it's titled Evaluation of Antioxidant and Antibacterial Potential of Honey, produced from stimulative diet-fed bee colonies. And basically, the core objective here is to explore how scientifically altering a honeybee's diet can fundamentally upgrade the medicinal quality of the honey it produces.
And, it's interesting because modern evidence-based science is really finally catching up to traditional healthcare systems on this front.
Right, like Ayurveda, right?
Exactly. If you look at Indian Ayurveda, honey has been like a prescribed medicine for millennia. Traditional practitioners haven't just used it to sweeten their tea.
No, they were using it for serious stuff.
Yeah, treating deep wounds, severe burns, dermatitis, even complex digestive disorders. They understood it had this functional power, even if they didn't have, a modern laboratory to map out the exact chemical pathways.
Well, if a natural substance is going to treat a deep wound, I mean, it has to be more than just sugar and water.
Oh, absolutely.
Looking at the ingredient list of pure honey from the study, the complexity is just staggering. We are talking about like a dense matrix of vitamins, minerals, proteins, enzymes, amino acids.
Flavonoids, phenolic compounds.
Yes, all of it. It's packed.
And the key thing to remember is that those compounds don't just exist in isolation, they function collectively.
Like a team.
Right, the phenolics, the flavonoids, and the specific enzymes, they operate synergistically. And that synergy is what physically generates the antioxidant, antimicrobial, and anti-inflammatory properties we're talking about.
But the quality of the honey isn't always the same, is it?
No, not at all. That's a critical variable that is so often overlooked. Honey is not a standardized manufactured product.
Right, it's made by nature.
Exactly. Its biological quality fluctuates wildly based on the floral origin, the geography, the climate, and, well, the foundation of it all, the nutritional status of the worker bee.
So let's look at the bee's internal machinery for a second, because this is fascinating. How does a bee actually take, raw nectar from a flower and turn it into this complex medicinal matrix?
So the transformation happens inside the bee's internal pharmaceutical factory, basically. It's primarily driven by the hypopharyngeal glands.
Which are located in their heads, right?
Yes, right in their heads. When a worker bee is healthy, those glands synthesize highly specialized enzymes, like glucose oxidase and diastase.
Okay, so they make the enzymes.
Right, and then the bee secretes these enzymes into the raw nectar as it's being stored and transported in their honey stomach.
So the honey stomach isn't just like a biological backpack for carrying nectar back to the hive.
No, not at all. It's an active biochemical mixing vat.
Wow.
The enzymes immediately begin breaking down complex sugars and triggering these chemical reactions that alter the pH. They even generate compounds like hydrogen peroxide in there.
Which is crazy to think about happening inside a tiny insect. But to run that internal factory, the bee requires specific raw materials, right?
Exactly. They get their carbohydrates from the nectar itself, but they get the essential amino acids and proteins required to actually build those enzymes from flower pollen.
So nectar is the energy, pollen is the building blocks.
You nailed it!
So imagine like a high-end pharmaceutical factory where the supply chain of those raw materials suddenly collapses. The workers can't synthesize the medicine anymore.
Right, they just don't have the parts.
Yeah, they might try to piece things together with whatever scraps they have lying around,
Right.
but the final product is going to be incredibly weak. And that is essentially what happens to a bee colony during what beekeepers call a dearth period.
Yeah, the dearth period is tough. It occurs when natural floral sources just dry up.
Like during a harsh, dry summer.
Exactly, or during specific seasonal shifts. The nectar stops flowing, but more importantly, the pollen disappears. So the colony faces severe nutritional stress.
because they can't make the enzymes.
Right. Without the amino acids from pollen, the worker bees cannot maintain those hypopharyngeal glands we talked about. Enzyme production just plummets.
Making the honey weaker.
Exactly. The resulting honey they manage to produce during this starvation time lacks that dense bioactive chemistry that makes it medicinal.
So the researchers in this study, they looked at that exact problem. And they designed an intervention for Apis mellifera colonies, which are just Western honeybees.
Right.
They wanted to know if we artificially feed the bees a specially formulated stimulative diet during this dearth period, can we actually rescue or even improve the antioxidant and antibacterial properties of the honey,
Which is a brilliant proactive shift in apiculture.
rather than just letting them starve?
Exactly. Rather than letting the colony simply endure the starvation period and, accepting a chemically inferior harvest, the researchers formulated a nutritional supplement to replace the missing pollen.
To keep the factory running.
Yes, to keep the bees' internal factories running at full capacity.
And the recipe they used for this stimulative diet is super interesting. It's not just sugar water. They created these actual nutritional patties.
Yeah, it's a very dense formulation.
Right. They made them out of defatted soybean flour, parched gram, brewer's yeast, protein hydrolysate powder, sugar, and a little bit of honey mixed in.
It's heavily focused on protein. The defatted soybean flour, the brewer's yeast, and the parched gram, they are really doing the heavy lifting here.
Giving them the amino acids.
Right. They provide this massive infusion of proteins and essential amino acids to substitute for that absent pollen.
Which keeps the colony healthy.
Exactly. It sustains the colony's brood rearing and maintains the physiological health of the adult worker bees.
But wait, here's a practical question I had when I was reading this. If the bees are instinctually wired to seek out blooming flowers for pollen, do they actually realize a patty of soybean flour is food?
That's a great question.
I mean, if it doesn't look or smell like a natural flower, how do you get thousands of stinging insects to just eat it?
Well, that is the genius of the methodology they employed. It's known as the top bar method.
Yeah, how does that work?
So the researchers wrapped these nutritional patties in butter paper and punctured them with little small holes.
Like poked holes in the paper.
Yeah, exactly. And then they placed these packets directly inside the hive, right on the top bars of the frames where the bees are actively working.
Oh, I see.
And the ambient warmth of the hive, combined with the formulation itself, causes the nutrients to slowly seep out through those punctures.
Oh, so it mimics the slow, steady supply of nutrients they would normally get from foraging.
Right, rather than just dumping a pile of powder in front of them, which they might just ignore or throw out.
That's incredibly smart.
Yeah, the nurse bees naturally investigate the seeping material, they recognize the sugar and protein content, and they begin consuming and distributing it throughout the colony.
So they feed it to everybody.
Exactly. And to measure the impact of this, the researchers monitored the hives over a full year.
Taking samples along the way.
Right. They took baseline honey samples before the diet intervention, when the bees are nutritionally stressed, and then they took samples after the prolonged feeding period to compare the biological activity.
And the first major test of that biological activity focused on antioxidants, which they used 3 different chemical assays to evaluate the honey.
Yes, the ABTS assay, the DPPH assay, and the FRA key assay.
Okay, right. These acronyms can feel totally impenetrable to anyone outside a lab. So, in plain English, what is the physical mechanism these tests are actually measuring?
Sure, let's break them down by the chemical principles they test, starting with ABTS and DPPH. Both of these are basically designed to measure a substance's ability to neutralize free radicals.
And just to clarify for you listening, a free radical is essentially an unstable, highly reactive molecule that is missing an electron, right?
Perfectly stated. And because it's unstable, it desperately tries to steal an electron from anything nearby.
Which is bad news for our bodies.
Yeah, in a human body, that means tearing into healthy cell membranes or mutating your DNA.
So an antioxidant is a molecule that steps in and neutralizes that threat.
Yes. An antioxidant is stable enough to basically donate an electron to the free radical without becoming unstable itself.
It takes the hit.
Exactly. It halts that chain reaction of cellular damage. So the ABTS assay uses a specific chemical radical to test how well the honey can donate an electron.
In the DBPH one?
DBPH operates on a very similar principle, but it uses a dark purple radical compound.
Oh, so there's a visual element.
Yeah, it's really
Yeah.
cool. When the antioxidants in the honey successfully donate electrons and neutralize the DPPH radicals, the solution physically changes color from deep purple to pale yellow.
Oh, wow, that's wild.
Yeah, and then a spectrophotometer measures that exact color change to quantify the scavenging capacity.
Okay, so that covers free radicals. What about the third test, the FRAP assay?
Right, FRAP stands for ferric-reducing antioxidant power. Instead of measuring free radical scavenging, it tests the honey's ability to reduce iron.
Reduce iron. How does that work?
It basically measures how effectively the antioxidants in the honey can donate an electron to an oxidized iron compound, which changes its chemical state.
Oh, okay, so why do they need 3 different tests?
Well, by using all 3 assays, the researchers get a comprehensive kind of 360-degree evaluation. If a sample scores highly across varying chemical mechanisms, it confirms the substance is a robust, multifaceted antioxidant.
Okay, so let's look at the data, because the results paint a very clear picture.
Hmm.
In the DPPH radical scavenging assay, the pre-feeding honey, the stuff produced while the bees were stressed, scored a scavenging grade of 20.55%.
Right.
But the honey collected after the bees were fed, the soybean and gram diet, jumped to 29.01%.
Which is a really notable increase.
Well, I wanted to ask about that. Is a jump of roughly 8.5% actually meaningful in a biological context?
Right.
I mean, on paper, moving from 20 to 29 doesn't sound like a landslide victory.
I get why it looks that way, but in the context of biochemistry and unrefined natural products, an absolute increase of that magnitude is highly substantial.
Really?
Yeah, it translates to millions of additional free radicals being neutralized per milliliter of honey.
Oh, wow. Okay, millions. That puts it into perspective.
Exactly. And the ABTS assay showed a similar trajectory. The pre-feeding honey measured around 88 micromoles per liter of neutralizing power.
And the post-feeding.
The post-feeding honey reached a maximum of over 91 micromoles per liter.
Okay, so the radical scavenging definitely improved. Did the FRA assay reflect that same surge in power for the iron reduction?
So the FRAP values did increase, moving from 0.296 to 0.396 milligrams per 100 grams.
Okay, so it went up.
It did go up. However, the researchers noted that strictly from a statistical standpoint, this specific variance in iron reduction wasn't classified as highly significant.
Ah, gotcha. So it trended up, but maybe not enough to be definitive on its own.
Right. Yet the aggregate trend across all 3 methodologies definitively proved that the stimulative diet, likely the fatted soybean flour and the parched gram, significantly enhanced the final product's free radical scavenging capacity.
So bringing this back to you, the listener, if I am consuming this biohacked honey, what is that extra scavenging capacity actually doing inside my body? Why should we care?
Well, it acts as a direct defense against oxidative stress. Going back to those free radicals we discussed, when your body accumulates too many of those unstable molecules, which biologists refer to as reactive oxygen species,
Right.
and you lack the dietary antioxidants to neutralize them, the result is widespread cellular damage.
It's essentially like internal rust, isn't it? The oxygen is degrading the cellular structures over time.
That is a perfect analogy. That internal rusting process is a primary mechanism driving chronic, long-term health failure.
Say what kinds of failure?
Oxidative damage is heavily implicated in cardiovascular disease, the mutation of cells leading to certain cancers, insulin resistance and diabetes, neurodegenerative disorders.
It's a laundry list of terrible things.
Yeah, and just the overarching biological process of premature aging itself.
Wow.
So by improving the nutritional intake of the bee, we drastically upgrade the antioxidant profile of the honey, turning it into a far more potent intervention against that internal degradation.
That's incredible. Protecting cells over the course of decades is obviously vital for long-term health. But let's look at the other side of the study. What happens when the body is facing an immediate, acute threat?
You mean bacterial infections?
Exactly. We're dealing with an escalating global crisis of antimicrobial resistance right now.
Yes. The severity of the antimicrobial resistance crisis really cannot be overstated.
Superbugs.
Right. Pathogens are mutating and evolving defenses against the conventional antibiotics we've relied on for the last century. We are rapidly approaching a post-antibiotic era where common infections could become lethal again.
Which is terrifying.
It really is. The medical community is desperately searching for alternative antimicrobial substances, or complementary ones, that bacteria cannot easily adapt to.
And honey is uniquely positioned for this, right? Because it doesn't just rely on a single mechanism to kill bacteria. It hits the pathogen from multiple angles simultaneously.
Exactly. That multifaceted attack is why bacteria struggle so much to develop resistance to it. First, you have the physical properties of the honey itself.
Like the sugar content.
Right, it has exceptionally high osmotic pressure. Because it's supersaturated with sugar and has very low water content, it creates a severe osmotic gradient when it encounters a bacterial cell.
Meaning the honey physically draws the water out of the bacteria.
Yes, the water inside the bacterial cell rushes out to balance the concentration, causing the cell membrane to basically collapse inward. The pathogen is fatally dehydrated.
So it dries them to death. What's the second angle?
Add to that the low pH. Honey is quite acidic, which denatures the proteins of most infectious bacteria, so they can't survive the environment.
And then there is the chemical warfare component we mentioned earlier, the hydrogen peroxide.
Yes. Thanks to that glucose oxidase enzyme produced in the bee's internal factory, the honey generates a steady, slow release of hydrogen peroxide when it comes into contact with moisture.
Like the moisture found in a human wound, for example.
Precisely. The hydrogen peroxide acts as an oxidative bomb, degrading the bacterial cell wall. And furthermore, there are diverse phytochemicals derived from the plant nectar that actively disrupt the bacteria's ability to communicate and multiply.
It's like a four-pronged attack.
Yeah.
Dehydration, acid, peroxide, and phytochemicals.
It's a nightmare for a bacterial cell.
So the researchers put this multi-pronged arsenal to the test against two severe medical pathogens, Escherichia coli, or E. coli, and Salmonella enterica.
Right, and these are not minor bugs. They cause aggressive systemic infections and are notoriously difficult to eradicate once they really take hold.
So how do they test it?
The experimental setup basically involved exposing these pathogens to various concentrations of both the pre-feeding and post-feeding honey.
Good.
They pushed the concentration all the way up to 800 micrograms per milliliter to really observe the inhibition of the bacterial growth.
And the results here were just stark. I mean, the pre-feeding honey, the stuff harvested when the bees were lacking the necessary protein to synthesize their defensive enzymes,
Right.
it showed practically no considerable antibacterial activity at the tested concentrations. The E. coli and Salmonella were largely unaffected.
Which is a very sobering reality regarding the commercial honey sitting on grocery store shelves right now.
Yeah, that's what I was thinking. If it was harvested during suboptimal conditions like a dearth period, it might be little more than a sweet syrup with negligible medicinal value.
It really highlights the massive variance in natural products.
But the post-feeding honey, the honey produced after the colony had been saturated with that soybean flour and graham diet, it behaved entirely differently.
Completely
It
different.
exhibited exceptionally high antibacterial efficacy.
Yeah, it was remarkable.
So what does that bacterial defeat actually look like in the Petri dish?
So at that 800 micrograms per milliliter concentration, the post-feeding honey aggressively inhibited the proliferation of the bacteria.
Because of that four-pronged attack.
Exactly. The E. coli cells are simultaneously dehydrated by the osmotic pressure, they're bathed in an acidic environment, and they're structurally compromised by the enzymatically produced hydrogen peroxide.
Wow.
The robust chemical profile of the post-feeding honey just completely overwhelmed the bacteria's defenses. It showed profound activity against the E. coli particularly, and demonstrated significant inhibition of the Salmonella as well.
The disparity between those two samples perfectly illustrates the biological mechanism at play here. The diet literally gave the worker bees the precise amino acid building blocks they needed.
Right.
Their hypopharyngeal glands functioned optimally, secreting a superior volume and quality of enzymes into the nectar, resulting in a chemical matrix capable of dismantling dangerous pathogens.
It's beautiful, really. The study elegantly proves that the medicinal chemistry of the final product is inextricably linked to the physiological health of the insect producing it.
It's all connected.
Yep. By proactively managing the nutritional intake of the colony during environmental stress, beekeepers can ensure the honey actually retains its complex functional power.
It is the ultimate confirmation that you are what you eat, but extending to the species that manufacture our food.
Exactly.
A simple dietary upgrade of soy and graham transforms a struggling colony's honey into a legitimate weapon against antibiotic-resistant superbugs.
Which leaves us with a pretty lingering scientific question to consider for the future.
Oh, what's that?
Well, we now have hard data demonstrating that altering a single insect's diet dictates whether its byproduct can kill E. coli.
Right.
So if human activity rapidly alters global ecosystems, changing the natural foraging grounds and diets of countless animal species, what other powerful traditional medicines and natural biocompounds are we inadvertently degrading today without even realizing it?
Wow, that is a heavy thought to leave off on, an important one. Thank you for joining us on this deep dive. The next time you reach for that plastic bear in the pantry, hopefully, you view that golden syrup not just as a sweetener, but as a total marvel of biological